So today we're going to transition to the motor system. Now it's a somewhat artificial distinction because there's a lot of sensory signals in the motor system, and motor signals sort of propagate back into the sensory systems. But we're going to start with eye movements, and it turns out that a lot of the brain a substantial chunk of the brain is concerned with eye movements because it's so important to get the sensitive part of your retina onto an interesting target. So that's looking at something new. But today we're going to talk about stabilizing eye movements, and which is, looking at the same thing. So looking at the same thing, and what's the main reason for this? And, does it have any relevance to cognition? It does. So looking at the same thing is, the main reason there is that if something moves relative to your retina, you can't see it as well. So you get a lot of blur. You can just sort of imagine you got a bunch of detectors, they have a finite temporal response, and if you move something past them, they can't pick up the fine details. And so all animals that have a visual system have some equivalent mechanism to essentially stabilize vision. And this is, it's been built into your cell phone now. Like if you take a video, you're jerking all around, but the video comes out less jerky because it's got a system in there to try to cancel out movements of the camera or the eye. So I'm going to today talk about three main types of stabilizing eye movements, and then how some of the information that's collected in order to perform these movements, it goes up into the system, goes up into the cognitive system, it goes up into, for instance, the limbic system, where you have things like head direction cells that indicate what direction your head is pointing in a particular environment. So I'll start off with the first one. And I'll just do one board today, so I'll hopefully try to keep the diagrams a little more note-takeable. So we'll start off with number one. So number one is, I'll put it in green here. Number one is the vestibulo-ocular reflex. So this is the vestibulo-ocular reflex. And it's often just called VOR for vestibulo-ocular reflex. So what does this do? So this basically tries to cancel. It's a very specific thing. It can't do everything. It tries to cancel head rotations. So what this does is it, cancel head rotations. That's the goal of this. So what is the vestibulo-ocular reflex? It's basically when you rotate your head. So if you're looking straight ahead, and you rotate your head and continue looking straight ahead, you can, if you look in the mirror and do this, just move your head around and look straight into the mirror, and you'll see your eyes are just rotating around in your eye socket in order to keep your eyes straight ahead. It's a very high performance system, so I can look straight ahead and do something like about this fast, and my eyes are just going, and they're just sort of keeping my eyes looking stably at whatever I'm currently looking at. And the vestibulo-ocular reflex, it not only does sort of, left, right, up, down movements, but it rotates the eyeballs too. And so if you look in the mirror and just tilt your head, you can see your eyeballs rotating in the eye sockets. So they can rotate about 30 degrees. So like when you tilt your head like this, the world is actually still still straight. You have to go like to about 90 degrees before you get past the ability of your eyeballs to rotate. So this is a very fast system, and it uses the semicircular canals, which, like I said, should be called the circular canals because they're actually circular, and they've got a little bank of hair cells in there, and then the bank of hair cells are the hairs are embedded in a little gelatinous flap that's kind of like attached to the hairs, and the whole thing is filled with fluid. And the way it works is if you rotate that little circular canal, the water tries to stay in the same place and it pushes on the flap. So the flap will just get a little deflection with a rotation. So that's fundamentally how this system works. And so it can only really detect angular acceleration. So it doesn't detect sort of continuous rotation. So like if you get on a merry-go-round and go continuously around in a circle, the system can't detect it. It can only detect like a change in the angular velocity. So what does that mean? It means like now we got angular velocity 0, and then I rotate my head. There's at the beginning of my rotation, there's a sudden change in angular velocity. That's angular acceleration. And then as I continue rotating my head, there's no change. So it's really just detecting that initial change in angular velocity. So the change in angular velocity is the acceleration. Derivative of the velocity is the acceleration. So that's what it detects, and there's 3 of them. So there's another one that sort of comes out in this plane, and another one that sort of comes out in the vertical plane. And so there's 3 separate sets of hair cells that are sort of detecting the 3 rotations. So there's 3 possible rotations. You could sort of like rotate this way or rotate that way, or what was the last one? Or rotate, so I did this way and then that way and then that way. So you have three different planes of rotation that you can detect. Now, what happens if you rotate your head in some other plane? Because obviously you don't just go around with just sort of X and Y movements. So in general, a general head rotation is going to give a little bit of a signal from all 3. Because if you don't rotate in the horizontal plane, but you rotate a little bit off the horizontal plane, the horizontal semicircular canal is still going to give you a signal. So in general, you're going to get 3 signals for almost any head rotation. So the vestibulo-ocular reflex is, so it detects angular acceleration, and it's a feed-forward system. So it gets a signal. It gets a signal from the semicircular canals, and then it just does the eye movement. And we're going to see another stabilizing eye movement system. It is a feedback system where it gets a signal. That signal affects the signal that you're measuring, and then you measure the signal again. So it's a circular system. This one is just a feed-forward system. So it just detects the rotation, and then it just sends it to the eye muscles. And so what are the main parts of it? Well, first, there's the vestibular nuclei, which of course are square. Everything's square. So I usually put this in quotes because they're not just vestibular. So they get a ton of other input, as we'll see. So there's visual input coming in. There's auditory input coming in. There's a lot of other input coming into these. Really, the reason they're called the vestibular nuclei is because they get a direct input from the vestibular hair cells. So here's little synapse made onto that guy. There's the ganglion cell that's near the cochlear ganglion, and that guy comes in and synapses on these vestibular cells, vestibular nuclei cells, and because of that, that's why it's called the vestibular nuclei. But really, what they are kind of the general purpose nuclei for sort of maintaining posture, something like that. Because there's 4 of these nuclei, so there's actually 4 separate vestibular nuclei, and several of them go down to the spinal cord. And if something gets screwed up in your vestibular system, like you get an infection in there, you won't be able to walk, because it's just a high performance system constantly keeping my 160-pound body upright. I mean, I just imagine you got 160 pounds of meat sort of standing on 2 little poles, and for the most part, it doesn't fall down. So every moment of the day, this thing is busy telling all the muscles in the body to make sure, just don't topple over. So that's the vestibular nuclei, and they have a lot of other connections. So there's connections with the cerebellum. So there's a big connection that goes back and forth to the cerebellum, and there's a bunch of connections to the reticular formation, which are sort of motor structures that we'll talk about a couple of lectures from now. A lot of connections going in and out. And then it has, this huge output to the body. So it not only controls, eye movements, but, it's going all the way down to the spine. It controls your legs and your arms, a bunch of other stuff. Every time you move something, it's going to unbalance you. So, I mean, these things are so automatic, you don't realize. If you lift your arm up like that, what's happening is a whole bunch of other muscles are going to cancel out the fact that you've got some weight that's sort of hanging out over here. So anytime you do that, you can see my body is just subtly moving to my left, but I can't feel it. It just sort of does it. It just sort of does it all the time until you mess it up. And one way of messing it up is if you drink a huge amount of alcohol, it will actually get into the fluid inside the vestibular canals and change its density. And when it does that, the signal coming out will be a little bit off. And if you get really drunk, when you move your head, instead of your eyes canceling out the movement, it will overshoot or undershoot a little bit, and then it looks like the world moves, and then you get sick. So we'll talk a little bit later about why does the vestibular system always make you sick? It's kind of an interesting question. Okay, so that's the vestibular nuclei, and then what do they go to? They go to the oculomotor nuclei, which are also a square. They're not a square. These guys are the oculomotor nuclei. There's 12 of these. So there's 12 of these, but conventionally they're sort of divided into if you go and take a neuroanatomy class, there's the oculomotor nucleus, but that one actually controls four of the muscles for one eye. And then you have something called the trochlear nucleus, and that just controls one. And then there's the abducens, and that just controls one. So the oculomotor nucleus, it's just called the oculomotor nucleus, but it's got 4 separate parts in it for 4 different muscles. And so what are these muscles? So if we draw an eyeball here, so here's an eyeball looking out at you. So you basically got some muscles on the top of the eyeball. They're long. If you look at it from the side, like the eyeball looks like this, and those muscles are quite long. They kind of like stick out in the little space behind the eye, something like that, if you looked at it from the side view. But you've got basically 2 muscles that pull the eye sort of up and down, and you've got 2 muscles that pull the eye left and right. And then you have, if this is the nose here, you've got 2 muscles that are like this, that rotate the eyeball. So you got 2 separate ones that actually rotate the eyeball. So those are the 6 muscles for one eyeball, but then you got 2 eyeballs, so there's 12 total signals that have to come out. And if you look at the connections, the vestibular nuclei connect directly to these oculomotor neurons. There's a lot of other obvious connections, that connects to something here, and then that goes in too. But there's a direct monosynaptic connection, so the ganglion cell, vestibular nuclei, oculomotor nuclei, and back out. And so then these guys will go and synapse on synapse on all these muscles to move your eyeballs. And so that's the basic system. And so how does this system, gotta make sure we get all our eye muscles connected. There we go. So how does this system work? So it actually, these muscles, so the eyeball, it doesn't have much friction to it. So it's quite easy to move it around in the eye socket. The way these things work is the muscles are always on. So they're holding the eye in a particular position. And if you get sleepy, some of these circuits will start to turn off, and then your eyes will start sort of moving around, and the world will start moving. But normally, when you're awake, what happens is they're tensioned up, and they're holding the eyeball in a particular position. And you can tell that because if you push on your eyeball, you see the world will move. But as soon as you let go, it will go back to where it was before, because they're actually holding it in place. And so if you look at the way that what the response of these neurons looks like, I always like to, if you look at the input and output stages of the brain, that's where you've got like really hardworking neurons. And this attracted engineers, because they said, we actually figured out how these guys work. They're always on duty, Whereas some cognitive neurons, like, I'm distracted. I don't feel like, paying attention to that thing I'm reading right now. So those kinds of higher level neurons are often kind of a little lazy. The lower level neurons are not like that. They're always on, until you get sleepy. They're always on, and they're always like sort of, ready to go. And so these neurons, if you look at the firing rate of these neurons, they're nice big neurons because they've got a lot of they've got a lot of work to do. They might be zinging along at 400 hertz. So if you record from them and put it through an amplifier, put it through a speaker, you'll just hear they're just going, it's just like firing like 400 spikes a second, continuously holding the eye in some position. So these are oculomotor neurons. Oculomotor neurons. And so what does their firing rate look like? So if you're just fixating something, they're just blasting away. And then if you decide to look at something, and say you're in a muscle that is in the pulling direction that you want to look at. So, here's the eyeball, and say you want to look at something that's off to the side, then you need this abducens nucleus to pull the eyeball off to the side. And the way it works is it just gives the eyeball a little yank. So it's holding it, and then it gives it a powerful little yank, and then the eyeball starts to rotate like a ball bearing in the eye socket, and then it has to be stopped. And so then the other muscle on the other side gives it a little yank to stop it. And that happens in only like about a 50th of a second. So it is truly a high performance system. And so the little yank will appear as a little spike in the firing rate, and then it will level out to a new position, new eye position further off to the side, if this was the abducens. And then if you go the opposite direction, it'll actually give you a little depression before it goes. And so this pattern this pattern of oculomotor neurons is called burst tonic, because it has a little burst, and then it has some tonic that just sort of means continuous firing rate. So that's the final output of the and we'll see as we go into the system, these different parts of the signal come from different parts of the system, like the burst comes from a different place than this continuous signal. But there's been a lot of research into the vestibular system and optokinetic nystagmus system, especially by NASA. And so I always tell this story about why you don't want to be an astronaut. So turns out when they put astronauts into space, what happens? Well, they often get sick. And so NASA says, well, we'll make them sick on the ground. We'll pick out the ones who don't get sick and we'll send those guys up. But they're still at about 50-50. So the NASA can make anybody sick on the ground. You pour cold water in your ears, put you in a centrifuge, turn you upside down underwater, all different kinds of things. But it still turns out that most people, a lot of people still end up getting sick. And if you get sick on the space station, think about like the space station. So like down here on the Earth, if you sneeze, stuff gets sprayed out, and then gravity takes over, and then it eventually goes down. If you throw up in space, what happens is these little globules of acidic vomit fly through the air, and then they stick to all the circuitry, and it's just like a terrible sort of cleaning problem in the space station because of this. And so that's one of the reasons why NASA funded so much, work into the vestibular system, trying to figure out how does this work or how does it go wrong? Because certain signals are still there when you're up in orbit, like all the semicircular canals are still working. They work in zero gravity, no problem. But other parts of the vestibular system don't work. Okay, so that's time there, firing rate going this way. So that's the final output, and this is a very high performance feed forward system that basically just keeps the world still. And like I said, it's one of these things like you don't realize how good it is until it goes wrong. And if it goes wrong, if you ever get like an inner ear infection, man, you're in deep doo-doo. I mean, literally cannot get up out of bed without falling down because once this system stops working. But like I said, it's not just low-level stuff. All these signals from the vestibular system go up forward into the cortex and are involved in all different kinds of higher level sort of knowing where you are in the environment. Okay, so that's any questions on the vestibulo-ocular reflex? So it's a feed-forward reflex that basically figures out the angular acceleration, feeds it back in, and makes the eyes cancel out that head rotation. So that's the vestibulo-ocular reflex. Okay, we'll put a dividing line here. Okay, so now the second one is so number 2 is called optokinetic nystagmus. Optokinetic nystagmus. And nystagmus is a Greek word, and it refers to the movement of your head, like when you're listening to a boring lecture. I've occasionally seen students out there doing this, and it's like a slow movement like this, and then it goes, woo, yeah, okay, like pinch myself, yeah, so like then you sort of start heading off to this side, then boom. So that's the kind of movement that optokinetic nystagmus is. And so what does this do? This basically, so this guy was, canceling head rotations. So this is, but that's not the only thing that can cause the image to move. So there's other things that can cause the image to move, and basically what this thing does is it tries to cancel movements of the world. Cancel movements of the entire world. And so when does that happen? Well, that basically happens when you move around in the environment. You could, on a science fiction show, the whole ship could get hit and everybody's jerking around on the bridge, but normally what happens is you just moved. So if you move relative to the world, the whole world is going to move relative to your retina and cause some slip on the retina. So this guy is a feedback system because it's trying to use a visual signal to cancel a visual movement. And so what happens is you'll get your retina will pick up a visual movement that will feed through the system, try to cancel that visual movement, and then you'll have to measure the visual movement again to see if you canceled it correctly. And so this system is a little slower than this feed-forward system, which just takes a signal and just immediately feeds it back. It doesn't have to sort of measure how well we did. So it's a feedback system, and so where does this system come from? Well, it takes the input from the eye. It takes a visual input. So here's the retina in the eye, and that projects up to several nuclei shaped like a square. And so these nuclei are there's several nuclei in the pretectum. There's one called the olivary pretectal nucleus. There's a little ball shape, looks like an olive. There's olives all over the brain. So there's several different nuclei there. And then there's another set of nuclei called the terminal nuclei, which is sort of, or sometimes called the basal optic tract. So there's actually a separate tract that peels off the optic nerve that goes into these terminal nuclei. There's 3 of them in a mammal, and so basically, so here's a retinal cell, and so that retinal cell, comes in and projects to the pretectum and to these terminal nuclei, and this guy is the basal optic tract. It actually sort of peels off the main optic tract. So then neurons from this structure project to the vestibular nuclei. I'll just draw them again. So here's the vestibular nuclei, same as they were before. Nuclei, we'll put it in quotes because you can see it's getting a whole bunch of visual input as well. So here's these guys, and they go down to the vestibular nuclei. So we got, and then they go to the oculomotor nuclei, which we already had before. So here's the oculomotor. And then it goes out to the eye muscle. So this guy goes down here. There's 12 of these guys, and then this guy goes and synapses on that eye muscle. So how does this system work? So it's detecting what's called retinal slip. So what is retinal slip? So what it's trying to detect, that's just like what the oculomotor people call movement. So what you're trying to do, your goal is to have no movement relative to the world. And so if you pick up some movement, you've done something wrong. And so you've got to essentially take that movement, feed it through the system, and do whatever you need to do to the eye muscles to stop it. So I've just been talking about this abstractly, but what is it actually? So you can easily see somebody. It's hard to see this in yourself because you need movement. But if you get a friend, have them look out of a car that's not moving too fast and ask them to look at the ground. Just pay attention as closely as possible to the ground. And what you'll see is their eyes will track the ground for a while, and then they'll make a rapid saccade back. And then they'll track the ground for a while and make a rapid saccade back. And that's the nystagmus type movement that we were talking about. So if you look at the, say, say, horizontal eye position. So here's horizontal eye position. What will happen is you'll have occasional saccades, but what the eye movement will look like is it will just go smoothly along, and then until you get to the edge of, the edge of your eyeball, edge of your eye socket, and then it'll make a quick saccade back, and then track again like that, and then make a quick saccade back, and then track. When you see somebody doing it, at first you go, oh, that looks kind of weird, like their eyes are just going like, wee, wee, wee. They make this very characteristic look of tracking the world. It's a very general thing. So if you take something like a turtle, you just put a turtle down on the table, and then just move a big drum of paper around the turtle will do the same thing. It actually won't even use its eyes. It will often use its neck. It will just use its neck and just track the paper going around like that, and then make a quick neck movement back and track. So it's a very general thing. This present, frogs have it, turtles have it. It's optokinetic nystagmus. And so this retinal slip is slow because you're almost stationary. So it's slow. It's like about 1 degree a second. So basically, once the system is kind of like cranked up, it's basically tracking the world, and all it's trying to pick up is the little offsets of we went a little bit too fast, and so we got a little bit of an extra rightward movement. But the neurons that are sensitive to this are they're much slower than, say, neurons in MT that are detecting 10 degrees a second or 50 degrees a second. This is like a very slow movement relative to the retina. So it's just the error. So this guy is just the error. It's not the actual movement. It's just the error in your tracking. And so that little error in the tracking will be fed into the system, and then it will send a signal to the oculomotor nuclei that will sort of try to reduce that error. So remember, the eye is already moving, and so it's just like give a little extra signal to this thing that's already moving the eye smoothly. So it's already moving the eye smoothly. So this is a smooth eye movement, and this guy is a saccade. So that's optokinetic nystagmus. And so I say this every year. So this is the time when we've reached the point where we have to do the pigeon imitation. So if you've ever seen a pigeon, this is more appropriate for London, a lot of pigeons in London, but if you've ever seen like a shorebird or any kind of bird walking along the ground, it looks like they're bobbing their heads. But they're not actually bobbing their heads. So what they're actually doing, so if I can sort of imitate a pigeon, so they're sort of sitting here like this. Their eyes are on the side. A lot of birds have their eyes mounted on the side so they can see predators and other things. And so if they just walk with their not moving their head, the whole world is going to move relative to their retina a lot. And so what they do is they sort of sit, they're standing there, and they make a head saccade, and then they walk under their head like this. And then they make another head saccade, and then they walk under their head like this. So they're not actually bobbing their head. So if you film a pigeon from the side while it's bobbing its head, what's actually happening is you see the pigeon here, and there's its head. It's completely stationary. And then the body walks under the head, because it's actually sort of moving its neck back. And then it makes a head saccade to a new position. And then it makes another head saccade. So if you film its head, what its head is doing is just going like, it's just completely stationary. It's just going bump, bump, bump. And so why are they doing that? Well, it's optokinetic nystagmus, to cancel movements of the whole world. And so you can see it's kind of a more general thing. It doesn't just involve the eye muscles, because in the pigeon it's all the neck muscles. And the same thing is true of the vestibular system. It's not just the eye muscles. It's the neck muscles. In fact, the vestibular system controls the feet. It's kind of the whole body. And so in this case, it's using the neck muscles to stabilize the world to eye movements, to all continuous movements of the whole world. Now, the important thing about this system is that it's not super high resolution because it's just trying to sort of stabilize the average movement of the world. There's a bunch of crap in the world. Stuff's flying around. You got to kind of ignore that. The branch, a little leaf is wiggling in the wind. You don't want that. What you want is like, let's just average, sort of sloppily average the whole world and just say, what direction is most of the world going in? And this system can kick in cases like if you're in a train and the train next to you starts to move, this system will kick in. So how does it kick in? Well, you're sort of doing something, maybe reading a book or something like that, and this movement signal appears in your periphery. And your optokinetic nystagmus system looks at the whole thing. It looks at the train window and the little handrest and a little piece of dirt on the train window, averages the whole mess, and it says, I think the world's going that way. And so what will happen is it will kick in this system, and this system will start tracking the world. It will actually move your eyes to follow the average direction of the world. Now, why does the world always, move? Why does the whole world move? Well, it's just generally because you've moved. And so what this will do is this will give you the feeling that the world has moved. Like you'll get the feeling, if the train goes that way, you'll get the feeling that you're moving backwards. It can work up and down too. So if somebody puts like a garage door up and the person's not paying too much attention, you can get the feeling that you're going like down into the ground. So it's a powerful system that really gives you a feeling of self-movement. And what they've done at NASA, one of the things they used to do at NASA a lot is get a big drum with bright stripes on the walls. So you put these bright stripes on the walls like this, black and white stripes, and then you take a potential astronaut and sit them down in the middle of this drum like that. They're looking out at the drum, and then you start rotating the drum, and so maybe they're sitting on a chair here. So what happens for about 2 seconds, you get the feeling like I'm just sitting here on the chair. And then if the drum's going that way, after about 2 seconds, it just feels like, whoa, you're just going around. And then you look down, it doesn't look like anything's moving down there, and then you start to get sick. So it's a powerful system. It gives you a powerful feeling of self-movement when the whole world moves, and your optokinetic system kicks in and tracks it. And like I said, it's not a high-resolution system. It's just trying to sort of figure out, which way is the world moving. So that system is, like I said, it's present in all vertebrates, and its goal is to cancel these movements by making a smooth eye movement, sort of similar to the smooth eye movements that you can get from the vestibular system, although these were generally a little bit more violent movements because you move your head almost like a saccade, but you can also just move your head very slowly, and it'll work even if you're just sort of moving your head slowly. Okay, so that's the optokinetic system. There's one other thing I wanted to say. It'll come back to me. Okay, so there's a third system I know what it was. Okay, yeah, finally remembered. Yeah, so the one thing I said at the beginning was, why does the vestibular system make you sick? It's kind of, like if you go on a roller coaster or you go on something that whirls you around, like what happens when you whirl around a lot? So if you whirl around a lot, you can actually get the fluid continuously moving in your vestibular semicircular canals, and what it can do is it can push that little flap off to the side because the fluid is continuously rotating around, like if you just like sit on a little turntable and rotate yourself. And so then what happens is Normally the flap just sort of gets bent off to the side and then it quickly goes back. Like you make a sudden movement and it goes back, make a sudden movement and it goes back. But if you get it really bent off to the side, it takes a couple seconds to go back. And that's what accounts for, like if you get on a turntable, rotate around, get going really good, get the fluid going around and then immediately get off, that flap will suddenly go back. And as the flap goes back over a couple seconds, you'll get this like, whoa, this huge signal that says like you should really rotate the other way. And you'll sort of stagger off the turntable. So, but why should that make you sick? Why should something like that make you sick? And it turns out if you go into the brainstem and look at, where we are, so we're down in the medulla, and if you look right, so where are the vestibular nuclei? Remember there's like the sensory and the motor parts, there's the left and the right, and this is the more motor part, that's the more sensory part. And so if you look at where the cochlear nuclei are, and then just go behind them. So if we just go, behind cochlear nuclei, because these are sort of the same kind of a thing as the hair cells in the cochlea. So if you go behind, that's where the vestibular nuclei. So this is the vestibular nuclei if you're looking for it on a section. Vestibular nuclei. So they're just behind the cochlear nuclei. But there's an area that's right next to there called right next to the vestibular nucleus called the area postrema. So area postrema. And that's kind of the vomiting center. And so it's actually quite close to the vestibular nuclei. Now, why would that be? So what's the story that Marty can tell about that? Well, these, like I said at the very beginning, these are like super high performance neurons. No nonsense neurons. They're working like day and night, just like, hardworking neurons. They're sending out like 100 times more spikes than your average cortical neuron. Just constantly keeping the eyes and the body and everything all straight. If something goes wrong, like for instance you ate some kind of poisonous neurotoxic thing, these are going to be good detectors that something has gone wrong. And so that's probably why, because this is such a high performance system, if it starts to sort of go bad, the body can detect, now you probably ate something, time to, whatever that was, get rid of it, throw up. And so that may be why there's this association between sort of the vestibular system and throwing up, because it's a way that the body can detect that something poisonous has been ingested because it slightly changed the gain of the vestibulo-ocular reflex. And so that may be, that's what NASA is trying to cancel. One last thing about why you don't want to be an astronaut. So if you go up, here's the Earth and here's the space station. It's only 100 miles above the Earth. But it's above the atmosphere. And so what happens is, normally what happens is cosmic rays come in and they slam into the atmosphere and release, those are super powerful photons, and they release a shower of fundamental particles. But if you're above the atmosphere, you get nailed by the cosmic rays. And so when the astronauts are in space, if they just close their eyes, they'll see these little flashes, and these little flashes are cosmic rays slamming into their retina and like probably annihilating a retinal cell here and there and causing little showers of fundamental particles in their retina. And so when they're in the space station, what do? They sleep between the battery and the Earth. So if you go next to a battery, then the cosmic rays coming in slam into the lead battery and get used up, and then the cosmic rays coming in from this side, of course you still get a few coming in that way, but less. Yeah, so that's why you don't want to go into space. It's good for robots up there. It's not good for people. You go up in space, like after like a month or so, you lose about 10% of the bone density in your heel, and it never comes back because there's no gravity up there. So, yeah, so my advice. Don't go into space.
So then what does that do to like your eyes when you come back? Like, do you get any?
You get various, well, people that come back from space, especially if they haven't had any gravity for a while and they haven't had anything, they haven't had any signal in their sacculus and utricle, they have, quite a terrible time. They usually don't film it, but they're usually just kind of like sagged out in some chair someplace trying to not walk around. And yeah, it's just kind of hard on the system in general. It's better down here. Okay, so the last system, it's a kind of a version of optokinetic nystagmus, so we'll call it 2A. And this one is called smooth pursuit. And so it has sort of a similar kind of a goal, which is to use a visual signal to stabilize a visual object. So this was, use a vestibular signal to stabilize a visual object. This is the VOR, optokinetic nystagmus. It's OKN for short. So OKN is use a visual signal from the entire world to stabilize something on the retina. And this one is track a small object. So that's actually, it's at odds with this optokinetic nystagmus. So track a small object. Now, a lot of animals don't have this. So cats, for instance, don't have this. So if you take a little attractive thing and move it in front of a cat, move it back and forth in front of a cat, the cat will just sort of go dun, 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 dun. It'll make some rapid head movements, but it won't smoothly track it. So what does this system look like when you operate it? Well, it would be something like, I look at the top of that little marker cap, and I just track that around with my eyes. I smoothly track it around with my eyes. My eyes are following it smoothly and keeping it from blurring on the retina. So this only really happens in animals that have like a really high resolution retina, like a monkey or an owl can do something like this. But something like a cat, they're relatively low resolution retina compared to a primate, so they don't really bother with it. Now this is a little harder problem because in this case, all we have to do is just kind of figure out the average movement of the whole world. Here we've got to actually find an object, sort of segment that object from the background, and then just figure out the motion just of that object while ignoring the motion of the background, because you can see it's, when you do this, so, this is like bail on the rest of the world. So if you're tracking a small object, 99% of your visual field is going to be moving around. Bail on rest of world. So this one, if you do this one, it's got to somehow cancel optokinetic nystagmus, because otherwise this signal would see, like, I just saw the whole world move as you tracked that object, and so I'm going to have to somehow deal with that. And so this one is a little bit more. So you start off with the eyeball down here. And there's a retina, and you go up to the DLGN. So here's where you go up to the DLGN, and the DLGN projects to V1, V1, and V1 projects to V2, and V2 projects to MT, and MT projects to MSTD. Remember that there's several parts of MST. It projects to a part that's particularly involved in detecting moving objects. And so you've got to do a bunch of visual processing to sort of like figure out which object do we want to track. And there might be multiple objects moving past each other, and you want to just, like, I'm just paying attention to the marker and I'm not paying attention to my hand, which is also moving. So this goes down to the pontine nuclei, and even though they're called the pontine nuclei, they're effectively in the midbrain. And so MST projects down to the pontine nuclei, MT projects down there also. And that's a big source, we haven't talked about it yet, but that's a big source of input to the cerebellum. So these guys go to the cerebellum, and in particular to the flocculus, which is a part of the cerebellum. Not just the flocculus, but that's one of the big places they go. So the pontine nuclei go to the flocculus. And then this cerebellum, it projects to the deep cerebellar nuclei, but eventually, well, I'll just leave that out. It eventually goes to the vestibular nuclei. So, back to the vestibular. And like I said, it's not just vestibular. Obviously, they're getting input from MST. So it's a lot more than vestibular input. So it goes to the vestibular nuclei, and then from there we go down to the oculomotor nuclei, and then back out. Back out to the eye muscles. So this is a slower system. This is a slower system because it's got to go. There's a lot more stuff, and so it turns out, and you can easily test this to see how this is slower by compared to, say, vestibulo-ocular. So I can move my head about like this, and I'm pretty much keeping the marker stable. But if I try to track it around, I start losing it once I go about 1/3 as fast. And if I go a little bit faster, then it starts to blur. So this system, it's got a lot more feedback going in it. There's more stages to it. It's got to parse objects, do more complicated things. But this is important if you've got a really high resolution retina and you want to actually sort of track an object and be able to sort of identify a moving object. Now, there's one key point here, is that this guy, if we run that one, it has to cancel this one. So think about it. It's if you were in NASA and they moved the whole world, for about 2 seconds, 1 or 2 seconds, you could hold out, and then pretty much it just feels like you're moving. Now look at smooth pursuit. I'm smoothly pursuing this. I'm just following it, and the entire world except for the marker cap is moving, and I don't feel like I'm moving at all. It just completely cancels this whole system. So this signal is still coming in through the terminal nuclei. It's saying like the whole world is moving, but somehow presumably in the vestibular nuclei, through the cerebellum, somehow the signals are coming in and say like, now we're tracking now, and so cancel all that world movement feeling. So finally, when you think about these systems, I've described them. I'm done just in 1 minute. So I've described them completely separately. Like you got the vestibulo-ocular reflex that's canceling rotations, and then you got this optokinetic nystagmus. It's trying to average the whole world. And then sort of like, cancel out a background movement. And this one is like cancel this guy and just track a little small object. But you can do these things. These things are all happening at the same time. So, if I'm tracking a small object, and what if I just rotate my head at the same time? So I'm tracking the small object, I'm doing optokinetic nystagmus, and then I'm doing this guy too. This guy's like sort of at the same time, there's a signal superimposed from the vestibulo-ocular that's getting rid of all the head movements. So so it's quite a complicated set of sometimes conflicting signals that this whole system has to integrate and decide, what's the best thing to do. So it's like I said, the engineers were attracted to this because, partly because it works well, and so they were able to make a reasonable amount of progress. But like I said, this is very important as you go up in the nervous system to higher level areas that are involved in trying to decide where you are relative to an environment. Okay, so any questions at the last second about vestibulo-ocular, optokinetic nystagmus, OKN, and smooth pursuit? Looking at the same thing. So the whole goal is just try to stop the world from moving. It's very important for robots too, for robot vision. Some of the first robots didn't have something like this, and then the video coming out of the robot's completely useless because it's jerking all around. Okay, not too far over. I think we'll end the recording there, David.